morning. Hello, my dear students. Today we will get clear explanations about the glucose metabolism, that is glycolysis. So, before that, we define what is glycolysis. Actually, in glycolytic pathway, glucose is converted to form the pyruvate in aerobic conditions or lactate in absence of oxygen, that is in anaerobic conditions. Anaerobic conditions. The Greek word glycolysis means glycas means the sweet and lysis means the splitting. So sweet splitting, that is called the glycolysis. Now glycolysis, where it occurs? It occurs all the steps taking place into the cytoplasm. So this is the objectives of these topics, how recognize the glycolysis as the major oxidative pathway of glucose. Then list of main reaction sequence of glycolytic pathway, and in that reactions what are the rate limiting enzymes and how they are regulated in this process, that we will discuss here, and assesses how many ATP has been consumed or how many ATP has been lost in aerobic and anaerobic conditions, and also there are the different pyruvate kinase deficiency, hemolytic anemia, and unique nature in glycolysis RBC. So many things is here, but we are interested about the glycolysis process steps and also what is the significance of this process. Now, glycolysis, the significance of this pathway, it is the only pathway which is taking place into the cell of the body. Secondly, glycolysis is the only source of energy in erythrocytes. Moreover, during the strenuous exercise, when muscle tissues lack of oxygen, enough oxygen, then anaerobic glycolysis forms the major source for the energy production for muscles. So the glycolysis pathway may be considered as the preliminary steps before the complete of the oxidation process. And it provides the carbon skeleton for the synthesis of non-essential amino acids. So it can one important things. Non-essential amino acid can be derived from this glycolysis process, carbohydrate molecule glycolytic pathways from this pathway, and as well as glycerol part of the fat. So it will let how it will happen, we will see in that subsequent steps. So now the steps of the glycolytic pathway. In the step number 1, what happened? Glucose, this is the steps aerobic and anaerobic oxidation. Glucose is converted to form the glucose 6-phosphate. And in that cases, there is an enzyme which is called the hexokinase or glucokinase. Mind it, one molecule of ATP has been used or utilized in this process because ATP it will convert to form the ADP and PI. Secondly, it is a key regulatory enzyme, and the kinase it is also irreversible reactions. Hexokinase or glucokinase, what I have used the name, both two are the isoenzyme, you mind it, and these are the isoenzyme, the same in nature, and their Km value are different, but their affinity also to the substrate has also different, but it is both these are the isoenzymes. Now in the step number 2, what happened? Glucose 6-phosphate is isomerized to form the fructose 6-phosphate with the help of the isomerase enzyme, phosphoxoisomerase enzyme, and again this fructose 6-phosphate which is converted to form the fructose 1,6-bisphosphate with the help of this phosphofructokinase enzyme. Again, PFK, phosphofructokinase, is the rate limiting enzyme of glycolysis, and it is allosterically regulated enzyme, and the enzyme catalyzes the second phosphorylation step of glycolysis using a second molecule of ATP. It is an important key enzyme in this pathway. This is the phosphofructokinase. Now step number 4, what happened? Then the 6 carbon of fructose 1,6-bisphosphate, it will split to form the 3 carbon of glyceraldehyde 3-phosphate and 3 carbon molecule of the dihydroxyacetone phosphate. So total number of 6 molecule ATP is formed here, and ultimately in that case there is an enzyme that is called the aldolase. It will help to form the splitting of these two pathway, and now glyceraldehyde 3-phosphate it will again convert it to form the dihydroxyacetone phosphate with the help of isomerase enzyme, and same thing is happened dihydroxyacetone phosphate or DHAP. It will be converted to form the glyceraldehyde 3-phosphate with the isomerase enzyme. So fructose 1,6-diphosphate, we will convert from the two-way glyceraldehyde 3-phosphate. Step number 4, glycolysis. The 6 carbon of fructose 1,6-bisphosphate, which is cleaved to form the 3 carbon units, that is, one is glyceraldehyde 3-phosphate, another is dihydroxyestone phosphate. But here the enzyme that is called the aldolase. And once again, if you see glyceraldehyde 3-phosphate, it will be converted to form the dihydroxyestone phosphate with the help of isomerase enzyme. So this can also be converted to form the glyceraldehyde 3-phosphate, that is vice versa with the same enzyme. Now in the step number 5, what happened? Glyceraldehyde 3-phosphate is dehydrogenated and simultaneously phosphorylated to form the 1,3-bisphosphoglycerate with the help of NADH, NAD plus, and which will goes to the electron transport chain, and it will produce 3 molecule of ATP. In the step number 6, what happened? The energy 1,3-bisphosphoglycerate is tapped to synthesize 1 ATP molecule with the help of the bisphosphoglycerate kinase. And this is another example for the substrate level phosphorylation, and where the energy is trapped directly from the substrate without the help of complicated electron transport chain reaction. When the energy is trapped by the oxidation of the reducing equivalent such NADH, it is also oxidative phosphorylation. So this step number 6, it is also a reversible reaction. Now step number 7, what happened? This 3-phosphoglycerate, it will be isomerized again to form the 2-phosphoglycerate by shifting phosphate groups, third to second carbon atoms, and here the enzyme is phosphoglyceromutase enzyme, and this is also really reversible reactions. In step number 8, what happened? 2-phosphoglycerate is converted to form the phosphoenol pyruvate by the enzyme of enolase enzyme, and 1 molecule of water is removed from here, and a high energy phosphate bonds is produced. So the reaction is also reversible reactions. Now in the step number 9, in presence of oxygen during these reactions, phosphoenol pyruvate is first converted to form the intermediately to ketopyruvate, that is stable form. So unstable form to stable form, it will be converted phosphoenol pyruvate to dephosphorylated to form the pyruvic acid, that is pyruvate, with the help of pyruvate kinase enzyme. And 1 molecule of ATP generated during this reaction. This is again another example for the substrate level phosphorylation. So this is the total 9 steps of reaction. Glucose in the presence of oxygen, there is glucose converted to form the pyruvate. Now in the step number 10, what happened? This is step number 10 in case of anaerobic conditions. Pyruvate is reduced to form the lactate with the help of lactate dehydrogenase enzyme. We know that lactate dehydrogenase is an isomerase enzyme, isoenzyme. That is lactate LDH1, LDH2, LDH5, and these are all isoenzymes, and they have specific functions, specific roles, specifically LDH1, 2, 3, 4, 5. So it will be discussed in later again on another videos we will discuss here. Now to see how many ATP will be produced in this subsequent aerobic and anaerobic oxidations. First of all, if you see the aerobic conditions in presence of oxygen, there are the four steps where the ATP, 5 steps there are ATP will be consumed there. What are the steps? The steps number 1, hexokinase, there 1 molecule of ATP has been already it has been used, or that is one molecule of ATP already it can be utilized. And then step number 3, that is phosphofructokinase, 1 molecule of ATP minus, so total 2 ATP has been already consumed. And now ATP productions only in the 3 steps. Step number 5, where 1 molecule of NADH is utilized, and it can produce 3 molecule of ATP. And again 1,3-bisphosphoglycerate, there is one ATP utilized, and pyruvate kinase there also 1 ATP. So total number of ATP, if you see substrate level, in case of substrate level phosphorylation, there is 2 into 2, that is 4 ATP, and oxidative phosphorylation, there is 2 into 3, there is 6 ATP. So total 10 ATP has been used, and 2 molecule of ATP has been already utilized. So total 8 molecule of ATP in presence of aerobic conditions. So now in anaerobic condition, what happened? there is the same things happened. Only ATP conjugate two steps, hexokinase and phosphofructokinase. There are two ATP already utilized, and step number 6 and 9. There are two. What happened? One molecule of ATP already utilized in the step number 1 hexokinase, and another ATP already utilized, that is in step number 3 phosphofructokinase. And in step number 6 and 9, what happened? There are 1,3-bisphosphoglycerate kinase and pyruvate kinase is the enzyme. There are two ATP already used. That means that is oxidative phosphorylation 6 ATP and substrate level phosphorylation 4 ATP. Already we have discussed, and total number of ATP gain, that is 4 minus 2, that is the 2 ATP molecule. So these are the aerobic and anaerobic conditions where the ATP has been utilized or used. That we have been discussed here. That is two molecule of ATP. Already we have discussed that total number of ATP in anaerobic condition, 2 molecule of ATP has been generated, and in case of aerobic condition there is 8 molecule of ATP generated. Now to see the regulation part or regulatory part of the glycolysis. What is this? There are the two important enzymes, that is the hexokinase and glucokinase. We already discussed that it can regulate it. How it regulated? It is inhibited by the reaction product, and that is glucose 6-phosphate, which accumulate when further metabolized or metabolism of this hexose is reduced. Another enzyme, that is glucokinase. Both are the same type of enzyme, isoenzyme, but they are the different functions. Actually, glucokinase and hexokinase both have affinity separate to the substrate, and Km value also separated, and all glucokinase found in the liver only, whereas hexokinase found in all tissues. And glucokinase, it is inhibited indirectly by the fructose 6-phosphate and is indirectly stimulated by the glucose. How it reacts? This glucokinase, it can translocate and bind tightly with the protein, that is the glucokinase receptor protein or regulatory protein. How it happens? When GLUT or glucose uptake receptors, already we know that GLUT has there are so many types of GLUT 1, 2, 3, 4, 5, 7, and all these GLUT has some important properties, and they are found in different areas. Like the GLUT 1, that is it's found in RBC, brain, kidney, and it has main properties glucose uptake in the most cell. We discuss here the GLUT receptors, that is the GLUT2 receptors. That means it has low affinity glucose uptake in the liver, and glucose sensor in the beta cells of pancreas. So these receptor bind with this molecule protein, and then it can activate it. That is a glucokinase, and it will be again it can inactivate it from active form to inactive form. So this is the mode of action of when glucagon comes. This is the hormone. It comes from the pancreas of alpha cell. It binds with the receptor. Then ATP, with the help of adenyl cyclase, this through the cyclic second messengers, it will activate the protein kinase enzyme, and ultimately pyruvase kinase, it is inactivated form. It will again inactivate with the help of ATP. So in this way, how glucose molecule can be regulated here. So another thing is important thing we want to discuss here: pyruvate kinase deficiency, hemolytic anemia. Pyruvate kinase mutation may be lead to the altered enzymatic kinetics, so altered response to the activator, and decrease the amount of the enzyme or its stability. So this is the diagrammatic representations how enzyme acts here, and how it can be altered altered during this kinetics, and also decrease the amount of enzymes concentrations or its stability. So pyruvate kinase deficiency, hemolytic anemia can lead to this. These are the three important things. Long-term regulation of glycolysis. What happened? There are two important hormones which we are emphasizing: insulin, which comes from the beta cells from the pancreas, and glucagon comes from the also pancreas, islets of Langerhans, alpha cells. And both two colors used. One is the insulin. It can reabsorb the glucose molecule, and glucagon, it can enhance the glucose molecule. So this is the reaction sequence by which glucose can be regulated here. So insulin, glucagon, it is insulin is the induction. Induction means it can enhance the secretion, and glucagon is the repressor, means it can reduce or nullify the enzymatic action or the regulation parts. So this is all about the glycolysis process. And thank you. If you are the regulation of glycolysis, already we have discussed how it is regulated, and now the regulatory enzymes, that the glucokinase, hexokinase, both we have already discussed how they are regulated with the glucose molecules. And another enzyme, that is the pyruvate phosphofructokinase 1 and pyruvate kinase. And the regulatory mechanism, there is a short-term mechanism. If you see the allosterically, it can be bound or covalently modified with the enzyme actions. And slowly, if you are long-term, if you see, there is the induction and repression, both are the enzymatically regulated. So apply above these two way of mechanism, rapid and short-term or slow or long-term mechanism, and glucose or glycolysis can be regulated. So in the last part, we come here the significance of the glycolysis pathway. So if you see, there are so many significance. Why we want to see the pathway of glycolysis? Because it is the only pathway that taking place in all the cells of the body. Number one. Number two, glycolysis is the only source of energy in case of RBC. We have already discussed. And then during strenuous exercise, when muscle tissues lack of enough oxygen, anaerobic glycolysis forms the major source of the energy for the muscles. The glycolytic pathways may be considered as the preliminary steps before the complete oxidations. Moreover, the glycolytic pathway provides the carbon skeletons for the synthesis of non-essential amino acids as well as the glycerol part of the fat. And most of the reactions of the glycolytic pathways are reversible, and which are also used for the gluconeogenesis. What is the meaning of gluconeogenesis? Gluconeogenesis means the newly synthesized glucose from the non-carbohydrate. That means if you see fatty acid, amino acids, or a ketone body, from there glucose can be produced from non-carbohydrate to carbohydrate. So these are all about the glycolytic pathways, and this is the significance of the glycolytic pathways and their metabolism and consequence steps. So in the last part, what are the clinical importance of the glucose? Glucose is preferred the source of energy of the most of the body tissues. Brain cell derive the energy mainly from the glucose. And when the glucose metabolism is damaged, life-threatening conditions may occur. And a minimum amount of glucose is always required for the normal functioning of the physiological function of the body. Normal fasting glucose level, we know that 70 to 110 mg per deciliter, and after a heavy carbohydrate meal, in glucose, that means the after fasting, that is the postprandial glucose, that means average is below 140 mg per deciliter. That means 100 ml blood, it contains 140 milligram. So this is the importance or clinical importance of the glucose molecule. So already we have completed the total glycolytic pathway and their consequence or steps, and how many ATP or energy can be produced from glycolysis, and moreover the significance of glycolysis, why glycolysis is used or utilized. So that's all in these topics. So if you found this video helpful, smash that like button and hit the subscribe to see more. Or if you want to see more videos like this, make sure to subscribe. Thank you.